Top 5 foods that heal brain cells and restore memory. While you sleep right now, as you are engaged with this video, a subtle but significant process is occurring within your skull. Microscopic connections between your brain cells are gradually becoming less robust, and the accumulation of metabolic byproducts between neurons is likened to a backlog of uncollected refuse. The disquieting aspect of this internal phenomenon is its insidious nature. You will likely perceive no immediate discomfort or decline. This gradual fraying may persist for months, perhaps even years, before its presence becomes noticeable. Then, one morning, you might find yourself standing in your kitchen, opening a cabinet, and experiencing a disorienting blankness, questioning your purpose and the object of your search. This moment of cognitive pause, this brief but unsettling void, is not merely a symptom of aging. It is the outward manifestation of years of silent cellular attrition. However, a crucial element often goes unacknowledged. Your brain possesses an intrinsic repair mechanism. Every night, as you surrender to deep sleep, a sophisticated cleaning and rebuilding system is activated. This internal process diligently flushes away toxic waste, repairs damaged neuronal structures, and fortifies the intricate neural pathways that underpin your memories. This remarkable system is a scientifically documented reality, possessing considerable power. Yet, there is a critical caveat. This dedicated repair crew requires essential raw materials, specific nutrients, compounds, and building blocks that a substantial portion of the population, particularly those over the age of 60, are not adequately supplying. When these vital components are absent, the nighttime maintenance crew arrives at their station with inadequate resources, rendering them incapable of performing the necessary repairs. Consequently, what you consume in the hours leading up to sleep does more than merely satisfy hunger. It directly dictates whether your brain will engage in essential rebuilding and restoration or continue its silent deterioration. This discussion eschews any mention of supplements with flashy packaging, expensive powders, or purported miracle cures. Instead, we will focus on 5 specific whole foods, readily available in any grocery store, that provide the precise nourishment your aging brain requires to undertake its essential repair work during slumber. I am Dr. Thomas Reed, and over the course of this presentation, I will meticulously detail each of these 5 foods. We will delve into the scientific underpinnings of your brain's nightly repair processes, illuminate why this vital function begins to falter after 60, and elucidate the exact mechanisms by which each of these foods intervenes to rectify these issues. Some of the information I am about to present may be surprising, and some of it may even challenge long-held beliefs. However, rest assured that every assertion made today is substantiated by published research from reputable institutions. For individuals over 60, or for those who care about someone in that age bracket and harbor concerns about memory, mental acuity, or long-term brain vitality, this information could prove to be exceptionally significant. Therefore, I urge you to remain engaged, as we have a considerable amount of ground to cover. Before we embark on exploring these transformative foods, it is imperative to grasp a fundamental concept that many medical professionals regrettably fail to articulate to their patients. Your brain does not cease its activity when you sleep. Rather, it enters a different and arguably more active operational state. This is a common misconception. During the profound stages of deep sleep, your brain operates as one of the most metabolically active organs in your entire body, exerting more effort in certain respects than it does during your waking hours. The distinction lies in the nature of its tasks. While awake, your brain's focus is outward, processing sensory input, language, motor functions, decision-making, and emotional responses. It is in a mode of peak performance. However, upon entering deep slow-wave sleep, your brain shifts gears into a crucial maintenance mode. This is where the remarkable restorative processes take place. A decade ago, a groundbreaking discovery revealed the existence of the glymphatic system, which can be conceptualized as your brain's internal plumbing. During deep sleep, the interstitial spaces between your brain cells expand, increasing by as much as 60%. This expansion allows cerebrospinal fluid to surge through these widened channels, acting like a high-pressure cleansing system that flushes out the metabolic waste products that have accumulated throughout the day. Among the most concerning of these waste products is a protein fragment known as beta amyloid. This name may be familiar, as beta amyloid is the sticky plaque that characterizes the brains of individuals with Alzheimer's disease. However, a critical point often overlooked is that everyone produces beta amyloid daily. It is a natural consequence of neuronal activity. The issue is not its production, but rather the brain's capacity to clear it efficiently. In a young, healthy brain, the glymphatic system effectively removes beta amyloid during sleep. Unfortunately, after the age of 60, this system begins to decelerate. The intercellular channels do not dilate as widely, and the flow of cerebrospinal fluid diminishes. The cleanup crew is still present, but they are hampered by less efficient tools and a reduced supply chain. Consequently, waste material begins to accumulate night after night, year after year. This progressive buildup is directly correlated with memory impairment, cognitive decline, and ultimately neurodegenerative diseases. However, waste removal is only one facet of the nightly restorative process. Simultaneously, as the glymphatic system engages in its cleaning operations, another vital process unfolds: memory consolidation. During deep sleep, your hippocampus, the brain region primarily responsible for the formation of new memories, repeatedly replays the experiences and information acquired throughout the day. This repeated rehearsal strengthens the neural pathways that encode these memories, facilitating their transfer from short-term to long-term storage. This process, known as memory consolidation, is contingent upon the integrity and health of your synapses, the minuscule gaps between neurons where chemical signals are transmitted. Every memory, every learned skill, every recalled fact exists as a specific pattern of synaptic connections. During deep sleep, your brain selectively reinforces crucial synapses while pruning away those that are no longer relevant, effectively organizing and refining your mental archives. The critical takeaway here is that both of these nocturnal processes, waste clearance and memory consolidation, are biochemically intensive and demand specific resources. They necessitate healthy cell membranes, adequate neurotransmitter production, anti-inflammatory compounds to counteract oxidative damage, and the fundamental building blocks for neuroplasticity, the brain's inherent capacity for rewiring and self-repair. Post-60, the body's ability to generate and absorb many of these essential compounds naturally diminishes. Nutrient absorption from the digestive tract becomes less efficient, the liver's metabolic processes alter, and certain enzymatic pathways decelerate. Therefore, even with a generally sound diet, your brain may still be deprived of the specific nutrients required to fuel its nighttime repair operations. This nutritional deficit is precisely what the following 5 foods are designed to address. Each food targets a distinct aspect of nocturnal brain restoration, and collectively they furnish your sleeping brain with the comprehensive toolkit it needs to cleanse itself, rebuild neural connections, and shield itself from further damage. Our journey into enhancing brain health during sleep begins with the first food, one that might not immediately spring to mind when considering brain wellness, despite the remarkable scientific evidence supporting its efficacy: wild salmon. While the general advice to consume more fish is commonplace, the specific benefit of wild salmon for your brain during sleep operates through mechanisms that are often overlooked in mainstream health discourse, and it is deeply intertwined with the structure and function of neuronal membranes. Every one of your approximately 100 billion brain cells is encased by a membrane. This membrane is far more than a passive barrier. It is a dynamic, living entity that meticulously regulates the passage of substances into and out of the cell, governs the transmission of signals between neurons, and critically influences the efficiency of communication between brain cells. The primary constituent of these crucial cell membranes is a specific type of fat known as docosahexaenoic acid, more commonly recognized as DHA. DHA is not merely another fat present in the brain, it is the dominant structural fatty acid within your cerebral cortex, the outermost layer of your brain responsible for higher cognitive functions such as thinking, reasoning, and memory. It accounts for an estimated 15 to 20% of the total fat content of your brain. This is where the connection to sleep becomes paramount. During deep sleep, when your brain is actively engaged in memory consolidation and synaptic strengthening, the integrity of these cell membranes dictates the clarity and efficiency with which neural signals can traverse between neurons. If your cell membranes are rigid, degraded, or composed of suboptimal fatty acids, signal transmission becomes compromised, leading to noisy and inefficient communication. This is akin to attempting a phone conversation with a poor connection, where the information is distorted and memories are not formed or consolidated effectively. Post-60, the concentration of DHA in the brain naturally declines, a phenomenon documented in post-mortem studies which consistently reveal lower DHA levels in the cell membranes of older brains compared to younger ones. This decline is directly associated with measurable reductions in cognitive function and memory performance. Now, let us consider why wild salmon is particularly noteworthy. Wild salmon, especially sockeye salmon, is among the richest dietary sources of DHA globally. A modest 6-ounce serving of wild sockeye salmon delivers approximately 1,500 milligrams of DHA, a substantial dose. Crucially, unlike plant-based omega-3 sources, such as flaxseed or walnuts, which contain alpha-linolenic acid, ALA, a precursor that your body must inefficiently convert into DHA, the DHA present in salmon is preformed and readily absorbable. Your body can directly utilize this preformed DHA, transporting it to your brain without the need for complex metabolic conversion. However, DHA is not the sole reason salmon merits a prime position on this list. Wild salmon is also an exceptional dietary source of a compound called astaxanthin. Astaxanthin is the pigment responsible for the characteristic pinkish-red hue of salmon, produced by the microalgae that form a significant part of their diet. What makes astaxanthin exceptionally valuable for brain health is its unique property of crossing the blood-brain barrier, a highly selective gateway that controls the passage of substances from the bloodstream into the brain. While this barrier is vital for protecting the brain, it also means that many antioxidants consumed in food never reach brain tissue. Astaxanthin, however, is one of the rare exceptions. It effectively traverses this barrier. Once within the brain, it integrates directly into cell membranes, providing potent antioxidant protection precisely where it is most needed. It neutralizes free radicals directly at the membrane surface, thereby preventing lipid peroxidation that degrades DHA and damages cell membranes over time. Consider the implications for your sleeping brain. During nighttime repair processes, which are energy-intensive, oxidative stress is generated. If these free radicals are not neutralized, they can damage the very cell membranes that your brain is attempting to repair. Astaxanthin acts as an on-site protector, safeguarding the ongoing repair work. Scientific research supports these benefits. A study published in the British Journal of Nutrition demonstrated that astaxanthin supplementation in older adults led to improvements in cognitive function and a reduction in markers of oxidative stress in the brain. Furthermore, research published in the journal Marine Drugs indicated that astaxanthin reduced the accumulation of beta-amyloid plaques in animal models, directly supporting the glymphatic cleaning process. Lastly, wild salmon is an excellent source of vitamin B12. Absorption of vitamin B12 significantly declines with age, often due to reduced stomach acid production. Vitamin B12 deficiency is directly linked to brain shrinkage, not metaphorically, but literally. Brain imaging studies have revealed that individuals with low B12 levels exhibit measurably smaller brain volumes and a faster rate of brain atrophy compared to those with adequate levels. Vitamin B12 is essential for maintaining the myelin sheath, the insulating layer that surrounds nerve fibers and facilitates rapid and efficient electrical signal transmission. Degraded myelin slows down neural communication, leading to sluggish thinking, increased reaction times, and prolonged memory retrieval. By providing a highly bioavailable source of B12, wild salmon helps preserve the structural integrity of the neural wiring that your brain relies upon during its nighttime consolidation processes. To summarize, wild salmon is our #1 food because it delivers preformed DHA to rebuild and maintain brain cell membranes, provides astaxanthin that penetrates the blood-brain barrier to protect these membranes from oxidative damage during sleep, and supplies vitamin B12 to preserve the myelin insulation crucial for fast and clear neural signaling. These three distinct mechanisms, all derived from a single food source, directly support the vital functions your brain performs during sleep. It is crucial to emphasize the word wild in wild salmon. Wild salmon possesses a fundamentally different nutritional profile compared to farmed salmon. Farmed salmon typically contains significantly lower levels of astaxanthin, as these fish are often fed synthetic pigments rather than accumulating them naturally from microalgae. Furthermore, farmed salmon tends to have a less favorable omega-3 to omega-6 ratio, higher levels of environmental contaminants, and a lower overall nutrient density. Opting for wild-caught salmon, particularly sockeye or king salmon, represents the gold standard. Aiming for 2 to 3 servings per week provides a solid nutritional foundation. If one of these servings is consumed in the evening within 3 to 4 hours of sleep, you are effectively providing your brain with the necessary building blocks precisely when its repair shift commences. Moving on to our second essential food, we venture into an unexpected territory: tart cherries. If wild salmon represents the construction crew that rebuilds your brain during the night, then tart cherries act as the vigilant shift manager, ensuring that the crew arrives on time and is optimally equipped. Tart cherries possess a unique ability, unmatched by any other food on this list, to directly regulate the timing and depth of sleep itself, the very process that enables all subsequent nighttime repair activities. Specifically, the Montmorency variety of tart cherries is one of the few significant natural food sources of melatonin. This is not a synthetic supplement or an artificially created compound, but actual food-derived melatonin. Before you dismiss this as simply another sleep aid, allow me to elaborate on why melatonin is profoundly more important for your brain than you might currently realize. Melatonin is a hormone naturally produced by your pineal gland in response to darkness, and its primary role is to regulate your circadian rhythm, the internal biological clock that governs your sleep-wake cycles. However, melatonin also functions as a potent antioxidant, possessing a remarkable property known as cascade antioxidant activity. While most antioxidants neutralize a single free radical and then become inactive, melatonin, upon neutralizing a free radical, generates metabolites that are themselves antioxidants. This means a single molecule of melatonin can neutralize multiple free radicals in a chain reaction, rendering it exceptionally effective at mitigating oxidative damage within the brain. The significance of this cannot be overstated. After the age of 60, the body's natural production of melatonin declines dramatically. A 70-year-old individual typically produces roughly half the amount of melatonin compared to a 25-year-old, and sometimes even less. This age-related decline has two detrimental consequences for brain health. Firstly, it disrupts the architecture of deep sleep. You spend less time in the slow-wave deep sleep stages, which are crucial for glymphatic cleaning and memory consolidation. Sleep becomes more fragmented, with increased awakenings during the night, and the overall duration of restorative sleep diminishes, even if you spend the same number of hours in bed. Secondly, you lose the vital antioxidant protection that melatonin provides to your brain during those critical overnight hours. This is where tart cherries offer a powerful intervention. Research conducted at Louisiana State University found cytokines, fostering a cleaner and calmer environment for your brain's nightly restorative activities. A study published in the journal Food and Function reported that older adults who consumed tart cherry juice daily for 12 weeks exhibited significant improvements in memory, particularly in tasks involving visual and spatial memory. Another study in Nutrients found that tart cherry consumption reduced markers of oxidative stress and inflammation in the brains of aging animal models. A particularly fascinating detail is that tart cherries also contain meaningful amounts of tryptophan, an amino acid that serves as a precursor to serotonin, which in turn is a precursor to melatonin. Thus, tart cherries not only provide melatonin directly, but also supply the raw materials for your brain to synthesize additional melatonin. This offers both an immediate supply and a long-term boost to melatonin production. It is important to note that the form of tart cherries matters. When referring to tart cherries, we are specifically discussing genuinely tart varieties like Montmorency. Sweet cherries, commonly found in the produce section for snacking, do not contain the same concentrations of melatonin or anthocyanins. Therefore, opt for frozen tart cherries, dried tart cherries with no added sugar, or 100% tart cherry juice concentrate. The optimal timing for consumption is approximately 1 to 2 hours before bedtime, aligning the melatonin boost with your natural pre-sleep window. Furthermore, combining tart cherries with other foods on this list can create a synergistic effect that significantly amplifies the brain repair process, a concept we will explore later. Our third essential brain-boosting food is arguably one of the most underrated and accessible: eggs. These humble powerhouses have been a staple in grocery stores for decades, are incredibly affordable, and possess a wealth of scientific backing for their profound impact on brain repair during sleep. Specifically, we are focusing on whole eggs, with particular emphasis on the yolk, as it contains what I consider to be the single most critical nutrient for nighttime brain repair that is largely overlooked: choline. Choline is not a vitamin or a mineral. It is an essential nutrient that warrants its own classification. Your brain is remarkably reliant on it. Choline serves as the primary building block for acetylcholine, one of the most vital neurotransmitters in your entire nervous system. Acetylcholine plays a central role in memory formation, learning, attention, and the regulation of sleep architecture, especially REM sleep, which is crucial for processing emotional memories and creative problem solving. Following the age of 60, acetylcholine levels tend to decline significantly. This decline is so intrinsically linked to age-related cognitive impairment that the primary class of drugs used to treat Alzheimer's disease, known as cholinesterase inhibitors, functions by preventing the breakdown of existing acetylcholine. These medications do not generate new acetylcholine. They merely attempt to prolong the availability of what remains, underscoring the profound importance of this neurotransmitter for brain function. The critical issue is that the vast majority of individuals over 60 are not consuming adequate amounts of choline. A study published in the Journal of the American College of Nutrition revealed that less than 10% of the American population meets the adequate intake recommendations for choline, meaning over 90% are chronically undersupplying their brains with this essential nutrient for memory and overnight repair. The yolk of a single large egg contains approximately 150 milligrams of choline. The adequate daily intake for men over 60 is 550 milligrams, and for women over 60, it is 425 milligrams. Consuming 2 to 3 whole eggs per day can significantly contribute to meeting these targets. The choline in egg yolks is in the form of phosphatidylcholine, which is highly bioavailable and can be readily incorporated into brain cell membranes and utilized for acetylcholine synthesis. However, choline is not the sole reason eggs deserve a prominent place on this list. Egg yolks are also an excellent dietary source of lutein and zeaxanthin, two carotenoid pigments primarily associated with eye health, but increasingly recognized for their significant contributions to brain health. Research from the University of Illinois has shown that lutein accumulates in brain tissue, and higher concentrations of lutein in the brain are correlated with better cognitive performance in older adults. A study published in the journal Neurobiology of Aging found that adults with higher blood levels of lutein and zeaxanthin exhibited more efficient neural processing and superior memory performance. The mechanisms involved appear to include both antioxidant protection and anti-inflammatory activity within the brain, particularly in regions critical for memory and learning. Furthermore, egg yolks provide meaningful amounts of vitamin D, a nutrient deficiency that is exceptionally common in adults over 60, especially in northern latitudes and among those who spend limited time outdoors. Vitamin D is not merely a nutrient for bone health, it functions as a neurosteroid, actively involved in brain function. Vitamin D receptors are distributed throughout the brain, with particularly high concentrations found in the hippocampus, the very region responsible for memory formation and the primary site of memory consolidation during sleep. Research published in the journal Neurology indicated that older adults with severe vitamin D deficiency had more than double the risk of developing dementia compared to those with adequate levels. Another study in JAMA Neurology found a correlation between low vitamin D status and a faster rate of cognitive decline over extended follow-up periods. Eggs also offer complete protein containing all essential amino acids, including tryptophan, which, as previously mentioned in relation to tart cherries, is a precursor to serotonin and ultimately melatonin. Additionally, they provide vitamin B12 and folate, both crucial for homocysteine metabolism. Elevated homocysteine is a well-established risk factor for brain atrophy and cognitive decline. An adequate B12 and folate intake helps maintain homocysteine levels within a healthy range. It is important to address a prevalent concern regarding eggs: cholesterol. For decades, eggs were unfairly demonized due to their cholesterol content, leading to widespread recommendations to limit their consumption and avoid yolks. Many individuals over 60 still adhere to this outdated advice due to its long-standing promulgation. However, current scientific understanding reveals a different picture. The dietary cholesterol found in eggs has a minimal impact on blood cholesterol levels for the vast majority of people. The liver is responsible for producing the majority of the cholesterol in your blood, and when you consume more cholesterol from food, your liver naturally compensates by producing less. This regulatory mechanism has been substantiated by numerous large-scale studies. Notably, the 2015-2020 Dietary Guidelines for Americans removed the previous recommendation to limit dietary cholesterol to 300 milligrams per day, acknowledging that the scientific evidence did not support this restriction. More importantly, cholesterol is, in fact, essential for brain function. Despite constituting only about 2% of your body weight, your brain contains approximately 25% of all the cholesterol in your body. Cholesterol is a critical structural component of brain cell membranes and is indispensable for synapse formation and maintenance. Therefore, restricting your brain's access to cholesterol-rich foods while it is actively engaged in rebuilding synapses during sleep is counterproductive. In summary, whole eggs provide choline for acetylcholine production and memory consolidation, lutein and zeaxanthin for neuroprotection, vitamin D for hippocampal function, tryptophan for melatonin production, B12 and folate for homocysteine regulation, and essential cholesterol as a structural component for brain cell membranes. All these benefits are derived from one of the most affordable, accessible, and versatile foods available. Consuming 2 to 3 whole eggs daily, including the yolks, is a sensible and well-supported strategy for promoting brain health. If you have been discarding egg yolks based on outdated dietary guidelines, it is time to reconsider this practice, as your sleeping brain critically needs the nutrients they contain. Our fourth food, walnuts, is poised to fundamentally alter your perception of a pantry staple you likely already possess, due to its extraordinary mechanism of action. While each food discussed thus far has targeted a specific facet of nocturnal brain repair, wild salmon rebuilding cell membranes, tart cherries optimizing sleep, and eggs fueling neurotransmitter production and providing structural components, walnuts operate on a different level. They influence the repair process itself at a genetic level, modulating which genes within your brain cells are activated or deactivated during sleep. This direct genetic influence is a remarkable capability for any food. The primary mechanism behind this effect involves a compound found in walnuts called ellagic acid. Upon ingestion, ellagic acid is metabolized by gut bacteria into compounds known as urolithins, particularly urolithin A. Urolithin A has emerged as a significant area of research in aging studies over the past few years, and for good reason. It activates a cellular process called mitophagy. Mitophagy is your cell's quality control system for mitochondria, the powerhouses of the cell responsible for energy production. Your brain cells are among the most energy-demanding cells in your body, consuming approximately 20% of your total energy, despite making up only 2% of your body mass. This energy is generated by mitochondria. As we age, mitochondria accumulate damage.